Hello, my name is Halina Krzystek, and today I'll be presenting my master's internship. There are two parts, the first part being an evaluation of copy number variant calling algorithms for NCGENES here at UNC using exome sequencing. And part two is the application of machine learning clustering on microRNA data as a QA/QC tool in large cancer genomics projects here at the high throughput sequencing facility at UNC. So the first part is about copy number variant calling algorithms for exome sequencing. And I worked with the NCGENES study here at UNC. The NCGENES project aims to generate evidence for the use of exome sequencing as a first-line diagnostic tool in clinical genomics. The group is seeking to expand the diagnostic yield of its exome sequencing by identifying additional genetic variants, like CNVs, which may contribute to a clinical phenotype. My project goals were, 1, to explore the CNV-calling tools available for exome sequencing in a literature review, to evaluate their appropriateness for the NCGENES pipeline, and to compare the tool's performance on data from the 1,000 Genomes Project. Copy number variants are a significant source of human variation and have been found to be clinically relevant to phenotypes such as cancers and neuropsychiatric disorders like autism, schizophrenia, bipolar disorder, and epilepsy. CNVs are variants where the copy number differs from the normal copy number of 2 for a diploid organism. In this diagram, to the left you can see a heterozygous deletion of C resulting in a copy number of 1, and to the right, you can see a heterozygous duplication of C resulting in a copy number of 3. Homozygous deletions and duplications are also possible, as well as copy numbers greater than 4 up to 8 in non-tumor samples. It is also important to distinguish here that these are tandem duplications and deletions, and the algorithms I'll be reviewing actually cannot detect tandem versus non-tandem deletions and duplications. It cannot make that distinction. But both are possible. So first of all, what is exome sequencing? Exome sequencing is the sequencing of the exons or protein coding regions of the genome versus whole genome sequencing. The cost of sequencing in general is decreasing at an exponential rate since the first sequencing of the human genome in 2001. And ES is even less expensive than genome sequencing, making it a popular choice for patient testing. ES is also practical in that the majority of disease-causing variants have been found in protein coding regions. Previous methods to detect CNVs were microarray-based and typically had a resolution of 400 kilobases. Current methods are usually a combination of sequencing and microarray-based methods. However, it would be cheaper and faster to employ only ES. An additional benefit is that ES may provide a finer granularity in CNV detection than do microarrays, detecting CNVs as small as 1 or 2 exons. However, there are known shortcomings of ES CNV identifying algorithms, such as low sensitivity and a high number of false positives. There are many algorithmic approaches to detecting CNVs from genome sequencing. However, only one of those is applicable to exome sequencing, and that is the read-depth method. This diagram shows the basic concept behind the read-depth method. So you can see here, if you had a heterozygous deletion, you would expect half the reads from a normal diploid organism. Also, if you had a heterozygous duplication, you would expect about 50% more reads. However, this is under the assumption that nothing else is affecting read depth. And there are many biases in exome sequencing data that have to be accounted for that can impact the RD signal that are not indicating CNVs. For example, GC content bias, regions of poor mappability contributing to a decrease in read coverage, differences in the capture of target exons, and batch effects between samples. The read-depth algorithms that I reviewed used a 3-step process. First, the normalization of raw read count to account for biases like I listed. Second, segmentation of read count signal. Some algorithms use chromosomal windows of fixed length, while others adjust segmentation for confidence in calls, which impact the concordance between callers. And thirdly, they call copy number from likelihood ratios. In my literature review, I identified that most CNV detection tools were for somatic CNVs in tumor samples. The NCGENES2 pipeline, however, seeks to identify CNVs in germline samples, so non-tumor samples. Many of the review tools required matched tumor normal samples to compare copy number state, which did not apply to germline samples. There are far fewer tools available for the identification of germline CNVs using exome sequencing. While available for germline CNVs, Conifer and ZIM, two algorithms, use PCA and singular value decomposition algorithms for their normalization step, which require at least 100 samples that we do not currently have available in this phase of the NCGENES project. So I ended up selecting ExomeDepth, CNMOPS, and Codex, and ran them with their default parameters through Zimmer, which is a computational framework that allows you to compare output for ES germline callers. A brief comparison between the computational tools that I did select at the end of my literature review. Most of these tools differ in their assumption of read count distribution and how they account for other factors like differences in GC content, mappability, capture, and batch effects, and remove those biases. Secondly, they use different segmentation algorithms. Segmentation determines the length and position of a CNV. So two of the most popular methods that I found were the hidden Markov model and circular binary segmentation methods, which ExomeDepth and Codex use specifically. ExomeDepth also selects some of the given exomes to create a pooled reference set to which to compare copy number, whereas Codex and CNMOPS create Poisson Latin models, which they use as a comparison to call copy number states. I divided my results into 3 sections. First, characterizing the size and distribution of CNVs identified by each tool. 2, concordance between the 3 callers. And 3, concordance with a genome sequencing call set from the 1000 Genomes Project. The main takeaways of my analysis of CNV size and distribution was that the tool CNMOPS was unable to identify CNVs less than 1 kilobase in length, that ExomeDepth had the greatest range in CNV size, that ExomeDepth identified CNVs as small as 1 exon, and that Codex was able to identify CNVs as small as 3 exons, and that on average, CNMOPS and Codex identified larger CNVs than ExomeDepth. Looking at the concordance between these 3 callers, at the exon level, only about 25% of the exons called by ExomeDepth as having a CNV were confirmed by 2 other callers. Target exons were called as having a CNV if they had an overlap of at least 1 base pair with a CNV, as those would be ones that we are interested in investigating down the line. Here we have an IGV view of a deletion that was called in all 3 callers and the comparison to the read depth. So what we have here is we have 3 tracks for ExomeDepth, CNMOPS, and Codex. And you can see that they made a call here, and it was a deletion in all 3 tools. And over here, you see the reference genome. They called a deletion in the gene SERPB1, which is from a family of proteins known to be involved in RTKs and cell growth. There are actually 5 exons missing reads in this gene, 3 of which you can see here, that are also included in this track, which are the target exons that we sequenced. However, not all of the gene is missing reads. As you can see here, the last exon has reads. And it's interesting that the callers extend, CNMOPS and Codex extend their deletion for most of the gene. And ExomeDepth actually calls the deletion all the way to a neighboring gene, SERPG, which is in the same family of genes. So I think the takeaway here is that what we're trying to generate is a condensed call set for downstream analysts to look at. And even hundreds of calls is a lot to manually look at in a viewer like IGV. So we want to narrow down calls that we have the most confidence in before passing them down to downstream analysts. So to do that, we looked at concordance with a call set generated from genome sequencing. It is important to note that CNVs called from GS calling algorithms may have breakpoints that fall outside the target exon capture and therefore may be more difficult to pick up using exome sequencing CNV calling algorithms. So we see here in the middle that only 180 of the 1213 or 15% of target exons identified by the GS call set as having a CNV were identified as such by all three callers. ExomeDepth achieved the highest sensitivity with a sensitivity of 40.73%, a precision of 5.3%, and almost 9,000 false positives. Codex had a sensitivity of about 31%, a precision of 2.4%, and about 14,000 false positives. And CNMOPS had a sensitivity of about 30%, a lower precision of 1.1%, and 31,000 false positives. So that brings the question of what role could be implemented in the pipeline to capture the most of these true calls. Currently, the NCGENES pipeline employs only ExomeDepth as its CNV caller. I was investigating if there was any benefit to incorporating one or more additional callers into the pipeline. So a rule requiring a CNV call to be confirmed by all three callers is too stringent. It results in a sensitivity of only 15% capturing these 180 exons, target exons, gives you a precision of 8%, and still includes about 2,000 false positives. A second rule requiring a CNV to be confirmed by two or more callers is better, with a sensitivity of 37.5%, a precision of 3.1%, but still over 14,000 false positives. Finally, a rule requiring a call to be confirmed by ExomeDepth, which is already integrated into the pipeline, and at least one other caller gives a sensitivity of 35%, a precision of 6.4%, and 6,000 false positive. So requiring confirmation by ExomeDepth and at least one other caller gives the least trade-off in sensitivity, an increase in precision, and a decrease in false positives as compared to using ExomeDepth alone, as does the current pipeline. I further wanted to investigate the 631 missed exons that were missed by all three callers. Largely, those CNVs were on the X chromosome, suggesting that the pipeline may need to evaluate CNVs on the sex chromosome separately from those on the autosomes. I further characterized the CNVs by size and saw that most of them ranged from 100 base pairs to less than 200 kb, with a few outliers, and that was across deletions, duplications, and copy numbers called with a number greater than 4. A big question is, how can we distinguish true calls from the large number of false positives, which is a known limitation of CNV calling from exome sequencing? Comparison with the GS call set indicates that the best rule to implement in the pipeline is an ExomeDepth plus at least one other caller. This has the least trade-off in sensitivity, increases precision, and has a decrease in false positives compared to using ExomeDepth alone. However, my analysis indicates that the X chromosome may need to be evaluated separately from the autosomes. And a future direction in this project could be the application of machine learning to CNV detection. There's a program called CNLearn And as supervised machine learning, it requires a training set of at least 30 exomes with microarray data to generate a true call set. It can then take the input of multiple CNV callers and train a model to accurately detect true calls. If NCGENES decides to invest in generating a true call set from microarray data, then the application of CNLearn to train our model is promising. However, there is the limitation that a model trained on microarray data would call smaller true CNVs like those detected by ExomeDepth and codecs that were 1 to 3 exons in length, and that ES can detect. Potentially this machine learning model would call us false and we would lose that information. The second part of my internship was the application of machine learning on microRNA data as a quality analysis and control tool for projects at the high throughput sequencing facility on campus here at UNC. MicroRNAs are small non-coding RNAs that can regulate genes, for example, by degradation of messenger RNA or by blocking translation. And microRNAs have been identified as significant in multiple cancers, including breast, ovarian, glioblastoma, and leukemia. Machine learning clustering is a type of unsupervised machine learning, one that does not require a training set. It groups together samples by similarity in traits, and in our case, samples are grouped by similarity in microRNA expression. Two widely used methods for gene expression in cancer genomics are k-means and hierarchical clustering, whereas clustering on microRNA expression is still new. I based my hierarchical clustering pipeline on one for RNA-seq data and adjusted it for microRNA data. I designed my pipeline originally on some retrospective data for a large cancer genomics project and then tested it on some pilot data that came out recently from the sequencing facility. I used the program Cluster and then the program Java TreeView for visualization. So this is a view of Java TreeView, and to explain what we're looking at here, this is a heat map of hierarchical clustering. At the top, you can see a dendrogram or a tree diagram that groups similar samples with each other. The red in the heat map are microRNAs that are overexpressed in samples compared to the median, and the green are microRNAs that are underexpressed compared to the median. In the zoomed in window here, you can see the corresponding microRNA species to each line, and these are the species here. At the top, you can see the annotation of each sample, and you can see that these are tumor lung samples, specifically adenocarcinomas. So the pilot data was mostly from a project for clinical trials on lung cancer treatment. I clustered lung adenocarcinomas against lung squamous cell carcinomas. I was happy to see that the two formed distinct clusters, as seen here, that were largely homogeneous. The lung adenocarcinomas are here on the left and had a high node correlation of 0.46. The lung squamous cell carcinomas are on the right and clustered with a node correlation of 0.51. Further, there were 3 normal adjacent samples in this run, and I was happy to see that they clustered tightly together with a correlation of 0.59, and also that you can see they're at the edge of the lung squamous cell cluster, which is what we would expect given that these are normal tissue taken adjacent from tumor samples that are lung squamous. So that is what we would expect. On retrospective data, I noticed that certain types of cancers cluster well based on microRNA expression, and one of those was amyloid leukemia. So 90% of the amyloid leukemia samples in this run were in one cluster here with a node correlation of 50%. Glioblastoma also clustered well in retrospective data with 87.7% of those samples in a cluster with node correlation of 0.50. My observations from my runs on retrospective and pilot data were that amyloid leukemia and glioblastoma are two tumor types that cluster well based on microRNA expression. I also saw that lung adenocarcinomas and lung squamous cell carcinoma can cluster separately despite being from the same type of organ. And finally, that normal adjacents cluster separately from tumor samples. And this pipeline can be used as a QA/QC tool in that this has the potential to identify sample swaps that may occur at the sequencing facility and the mislabeling of samples. In addition, clustering can indicate samples of poor quality if they are not clustering well on microRNA data. I learned a good deal of what our group called forensic bioinformatics and the challenges in troubleshooting other people's code and these computational tools. From that, I learned the importance of documentation, documenting your own code, which I plan to carry into the future when I write my own code. I learned a lot about the application of computational tools to clinical and cancer genomics, and about the power and limitations of sequencing data, and how both computational tools and advances in sequencing technology can be used to improve health outcomes. So at this time, I would like to give an immense thanks to my mentors, Bradford Powell with the Berg Lab on his mentorship on the NCGENES project, and everyone in the NCGENES bioinformatics group, a huge thanks. I also want to thank Dr. Corbin Jones for his guidance and support on the microRNA project and the Jones Bioinformatics Work Group. These are my references, and at this time, I am happy to take questions or to give any further clarification. Thank you.